Hey, hello everybody, I am Jagannath. Welcome to our YouTube family Bio Gyan. I think you all joined the live section. So in this class we have discussed about your embryo. In the previous class we have discussed about your endosperm. So if you did not watch that last class, you may watch it. Okay. So in this class we have discussed about embryo and its basic concept, and the types of embryo has been seen, and how the embryo grow from a single egg cell to a totally fully developed mature seed or mature embryo. So what are the types of your embryo that are discussed in this topic? Okay, so basically in case of your embryo there has been two types of embryo, that is monocot and dicot. Dicots otherwise known as your eudicot. Eudicot means that multiple types of multiple cots have been present, that is multiple seeds has been grown at the time of your germination, so that it is called as your eudicot seeds. So seeds are protected by coats that is formed by your integuments. You may know the mature ovary has the part that is integuments, that does not take place in your sexual reproductive types. So it was just for protecting your seed. Okay, the seed coat is further divided into your outer layer, that outer coat that is known as testa, and the inner coat is known as tegumen. Okay, and hilum is a skull like outside of your seed where it is attached to your endocarp, that is fruit wall. Hilum कोटी था ना तुम्हारा endocarp पे था. Okay, how the hilum forms? Hilum form from the maturation of your seed. Maturation seed होला परे hilum रा formation होता है. सेटा केमिति होता है हमें next आगो को जानिबा. Okay, the micropyle is the small round structure in the next of your hilums where the pollen tube enter. During the process of your pollination and fertilization, the pollen tube enters through the micropyle side, you know. So this can also seen at your mature seed condition. If you have seen some rice or paddy seeds, you may seen the pulse like appearance at the upper part of your seed, that may be white or some yellowish like color, through which the germination first process takes place, that is your hilum side. Okay, so seeds have some embryonic axis. At the embryonic axis your premioles and radicals has been present. The premioles, which part? Premioles is the part through which the germination takes place, and it was the forming your shoot or stems of your plants. Okay, that is SAM. Shoot apical meristems are formed. That's the premioles have also premioles, which include shoot apical meristem of your developing seeds and leaf primordia. The leaf primordia, the premature stage of your leaf is leaf primordia. At the other end also the embryo axis has embryonic root, that is otherwise known as your radicle, and radicle is the apparent embryo. Okay, wait. Okay, radicle is not apparent in the embryo. That in case of your distal end, the root is simply root tip. Okay, the embryonic axis does not include your cotyledon. Embryonic axis में तुम्हारा cotyledon नहीं होता है. Okay, and the portion of your embryo between the cotyledon attached the point and the radicle, also known as your hypocotyle, means your below of your cotyledon. If the elongation found below of your cotyledon, that is your hypocotyle region, and if it at the upper of your cotyledon, that is your epicotyle region. Okay, some of your embryo may be lack of visible epicotyle. Epicotyle may not be seen because of your cotyledons are attached to the embryonic axis of your shoot tip. Okay, I think you understood. So ये टॉपिक गुड़ा को सेमित किसी डिफरेंस ना था, तो तुमने बुझी जाए तो इजीली. So we picturize this topic with picture, so you may able to understood topics very easily. So this is your seed and the outer coat. Okay, it is the seed coat, and we know that inner part of your seed coat is known as tegumen, and the outer part is known as testa. And you may see this type of structure in your mook dals and various type of various type of plants. So this is your hilum. Okay, so from which the hilum has been matured? The hilum is matured from your funiculus, which was present inside your, also in the ovules. Okay, the funicle is also a stalk-like substances that made for the othering the ovule inside your pod. Okay, and this part is your cotyledon. Okay, so below the cotyledon it is known as your hypocotyle region, and above the cotyledon it is your epicotyle region. Okay, so also the endosperm has been seen in this picture, that provides your nutrition to your embryo or mature seed. Okay, and radicle is the part through which your root has been developed, and primule is the part through which your stem has been developed. Okay, so I think you may understood. Okay, so I have also published this slide as prior, in which the flowers part has been developed into a mature structure, that is seed structures. So zygote forms into embryo, and polar nucleus sperms, that is form your endosperms, and these type of that is ovule will form your at the mature state seed, and the ovary wall will form your fruit, and funiculus form your hilums, and micropyle forms that micropyle. These structures has been seen in your seeds also very clearly. Okay, so you have to first know that what are the difference between the monocot seeds and dicot seeds. Okay, so in this picture you may able to know that, so you may able to know that there has been presence of your cotyledons. So the cotyledon in case of your monocot is single, which is otherwise known as your scutellum. This is your cotyledon, and it is otherwise known as your scutellum. Okay, we will clearly see in the next pictures. Okay, it is the seed of your corn, and it is the seed of your bean. So bean is basically dicot and corn is monocot, and which the scutellum which channels the nutrition to grow the embryo. But in case of your monocot and unicot, the presence of your have a single primules. The primules may be single primules which develops your seeds and seed primordia and shoot apical meristeme. Okay, here is your primules, like white color, white or green colors in the seeds. Okay, and sometimes white color in your monocot, like corns. Okay, and a hypocotyle embryonic stems below the cotyledons, and a radicle that forms your root. Here is your radicle which forms your root. Okay, primule forms your stem part and the shoot part of your plant, and radicle forms your root, and that includes radicals also includes also cotyledons. And the corn seed technically have your caryopsis, that means one seeded fruit. Corn have one seed in which the pericarp of your fruit is walled. Corn was covered by some walls, that is called as your pericarp. Okay, that is seed coat, and which fuse with your seed coat. Pericarp also fuse with your seed coat, that forms a covering to your seed. Okay, so this structure has been described in the picture. So in the two cotyledon in the eudicot, the two cotyledon in eudicot seed is connected to the rest of your embryo via a vascular tissue, that is xylem and phloem. What are the functions of your xylem and phloem? Xylem help into your water transport from source to sink to your plant, that helps for nutrition and transport of for the transport of nutrition and the metabolize micro component and macro component through the phloem tissue and phloems. Okay. In the endospermic dicot, the food reserves are stored in the endosperm. During the germinations, the two cotyledons therefore act as absorbing organ to take up a enzymatically release of your food reserved. So the foods which are reserved priorly, so that are enzymatically takes place in the part of your germinations, like tomato or tobacco. That is, tobacco scientific name is Nicotiana tobaccum, and tomato scientific name is Solanum lycopersicum. Okay, and pepper is Capsicum annuum. So like eight type of dicots plant गुड़ाक तुम्हारे कौन ना enzymatically activity होई कि that absorbing organ through organ गुड़ाक like act करन्ती तुम्हारे कौन ना cotyledon act करन्ती absorbing organ type रो. Okay, so these are seen in the dicot, and in the non-endospermic dicot means the the endosperm से भीतर नथिपो. So the triploid endosperm develop normally followed by your double fertilization. So double fertilization end product हम जानी चाहिए endosperm and zygote. So if the endosperm is not present, but the endosperm reserves are quickly remobilized to move into your developing cotyledon for storage. Endosperm act as your storage, and the two halves of your peanut seeds that are hypogea conditions or beans and split peas, like such species that split pea soups are divided into cotyledon loaded with food reserve. So এই পার্টটা হয়তো তুমি মানে বুঝি পার নথিবো. সো তুমি দেখিথিবো মটর দানা হেউ কিংবা ଆମର ମୁଗ ହେଉ କିମ୍ବା କୌଣସି ଗୋଟିଏ ସ୍ପେସିସ୍ରେ ଯେଉଁ ସ୍ପେସିସ୍ରେ କି ତୁମର କ'ଣ ନା ଡାଲି ଜାତୀୟ ଶସ୍ୟ ହୋଇଥିବ. ଓକେ, ସେମାନଙ୍କ ଭିତରେ ତୁମେ ଦେଖିବ ସେମାନଙ୍କ ଭିତରେ ସେଟା ଦୁଇ ଭାଗରେ ବିଭକ୍ତ ହୋଇଯାଇଥିବ ତା ସିଡ୍ସଟା, ଆଣ୍ଡ ସେଟା କ'ଣ ନା ବିକଜ ଅଫ୍ ୟର୍ ଦେୟାର cotyledon. Both cotyledons have use their nutritional purpose. Means କ'ଣ ତୁମେ କ'ଣ ଡାଲିଟାକୁ ଦେଖିଥିବ ଦୁଇ ଭାଗରେ ବିଭକ୍ତ ହୋଇଗଲେ ହାଫ୍ ପାର୍ଟ ଗୋଟେ ନ୍ୟୁଟ୍ରିସନ୍ ବି ଦୁଇଟା ଯାକ ନ୍ୟୁଟ୍ରିସନାଲ ପର୍ପସ୍ ପାଇଁ ହେଉଛି. ସୋ ଇନ୍ କେସ୍ ଅଫ୍ ୟର୍ ଯଦି ତୁମର କ'ଣ ନା ଏଣ୍ଡୋସ୍ପର୍ମଟା ନଥିବ, ସୋ ତୁମର cotyledon acts as your nutrition source for your seed. ଓକେ, ସୋ ଏ ପିକ୍ଚରଟା ଦେଖି ଦେଉଥିବ. ସୋ ଆମ ବାଦାମ କେସ୍ରେ କ'ଣ ବାଦାମର ଦୁଇଟା ସାଇଡ଼୍ରେ ଯେଉଁ ଏଇ ପାର୍ଟଟା ରହିଲା ଏଗୁଡ଼ା କ'ଣ ନା ତୁମର ଏ ଟୋଟାଲ୍ ପାର୍ଟଟା ହେଉଛି ତୁମର ନ୍ୟୁଟ୍ରିସନାଲ ସୋର୍ସ ଦ୍ୟାଟ୍ ଇଜ୍ ଅଲସୋ ଆଜ୍ ଏ cotyledon. ସୋ ବାଦାମଟା ତୁମର ଡାଇକଟଟା. ଓକେ, the presence of your this part is your premiole. Premiole means that has to grow the shoot apical meristem and shoot primodia. Okay, and in case of your peanuts, एटी करना से peanuts रे तुमर करना स्टेनिंग करथिला. को स्टेनिंग करथिला ना आयोडीन स्टेनिंग करथिला. आयोडीन स्टेनिंग रे कोन जानथिबो ना सुगर और कार्बोहाइड्रेट कंटेंट पार्ट गुड़ा को तुम कौन हम ना अदर ब्लू हम और ब्लैक कलर हम लाइट ब्लैक कलर हम. ओके, सो लाइक पीनट ईच ऑफ हाफ ऑफ योर पी रे देखी से पी मीन्स मटर गछ, पीनट्स मीन्स बादाम. ओके, in case of your pea that represents the nutrition filled cotyledon also, and the black coloration of your cotyledon is due to the iodine staining of your starch. Starch is a form of your stored carbohydrate or glucose, and most of your pea seed is filled with your two cotyledons. The tiny embryonic axis consists of your females at your top that is descending of your hypocotyls, and the radicle is the tip of your hypocotyl, and the seed coat is surrounded by entire seed. And this image by the following scientists. So हमें देखिथिबा मटर गछ देखिथिबो. मटर गछ जो फलटारे तुमर जो दुईटा भाग होई पारे सेटा जेहेतु से गोटे डाली जातीय शस्य आ से कोन ना से आयोडीन टेस्ट कले देखा तार ब्लू कलर पिगमेंट्स गुड़ा देखा मिलता है दैट इज योर प्रेजेंस ऑफ योर बिकॉज प्रेजेंस ऑफ योर स्टार्च ओके एंड बोथ द cotyledon that is hypocotyls that are acts as your nutritive source for our edible source and the seed coat also surrounded the seed which is your providing your protection from the outer environment. During the germination it protrudes into from the seed and forms the progeny or seedlet. Okay, because of your seed have a food reserves to fuel the germinations, they have also a nutritive food source for the peoples also. सेटा मु कहली तुमको आगरो. Okay, the starting of your nutrition content seed crops such as beans can use for increase the nutritive value of your plant using biotechnology. So it is the application of your biotechnology so that the cotyledon has been cotyledon needs naturally nutritive functions. So in case of your genetically changes in the cotyledons that may be other more nutrition value for the people also. Okay, so the following scientist is working on the combat to world hunger by the manipulating the plant in and improve into their nutritional quality. For example, her researcher in the bean is looking. So ये scientist से कौन ना following discovery also करथिले research करथिले. Okay, in case of your monocot seed कौन ना the seeds are most complex and in monocot family seeds of monocot रे complex होता है. काहे कि complex होता है ना due to presence of your single cotyledon that is your scutellums. Okay, monocots are basically the like Poaceae family species. Okay, so मैं जानती हूँ rice, wheat, these are monocots and cyanoderms ये species गुड़ा कौन ना monocots होते हैं. Because why? That monocots include the corn and wheat that are highly specialized that the testa and tegment that is your outer part and inner part of your seed coat are fused. Why the monocots are complex? Because that both the seed coats are fused each other and the fruits is caryopsis. Caryopsis means that grain formations. A single seed of fruit which is formed and the fruit wall is pericarp. So means कौन ना monocot case single seeds formation में होती है and the outer covering that covers the seed is your pericarp. So if you see the rice seed, the covering coated the seed is your pericarp. After we discharge and emasculate the seed coat that is pericarp of your rice, then it was edible. Okay. And thus not only the one or two layer of your seed coat has been fused but the seed coat is fused to form the pericarp. Okay, the single cotyledon is known as your scutellum and scutellum has also vascular connection with your rest of your embryo. एम्ब्रियो के साथ तुम्हारा क्या ना अटैच होकर रहता है तुम्हारा स्कूटेलम. ओके, द लार्जर इनर लेयर ऑफ योर एंडोस्पर्म दैट स्टोर्स द न्यूट्रिशन इज कॉल्ड एज योर starchy endosperm. Why it is called as your starchy? Because of your contents of your stored carbohydrate content, sugar, glucose content inside of your endosperms and the thin outer layer of your endosperm which is the single layer cell that is so known as your alluron. Why it was known as your alluron? Alluroning content present inside your endosperm. Okay, upon the germination the enzymes are secreted by your alluron containing some enzymes that degrade it and forms as your stored that degrades also your stored product that is like a carbohydrate, protein and lipids products and absorbed by your scutellum will be absorb the content degraded by your alluron and via the vasculated starts and developing your embryo. Therefore the scutellum can also seen the absorptive organs and not the storage organ. scutellum act करता है कैसे ना तुम्हारा absorptive organ की तरह. and the root tip is protected by a sheath like structure that is called as coleorhiza. The root was protected by or sheath like structure that is coleorhiza. When this radicle has formed, radicle also coated by your radicle form at the mature state, that is root tip formations that incorporate into your soil and functions as your source of your water and nutritional value from your soil. So coleorhiza is the root tip that will help to protect the root tip from your denaturations, and similarly the coleoptiles encase the premioles also for the shoot tips. शूट टिप को भी कोलियोटाइप कवर करके रखता है. ओके, सो दिस वाज द सीड स्ट्रक्चर ऑफ योर मोनोकोटिलेडन्स, monocot seed. Okay, in case of your monocot seed, that is presence of your radicals and premioles and the coleoptile that covers your one second. So the coleoptile and coleorhiza both are present to cover your root tip. In case of your radicals and root tips, that is your coleorhiza, and in case of your cotyledon and your premioles, it is your coleoptile. Okay, and endosperm has to provide the nutrition, and seed coat was covered the whole seeds, and it was fused with your pericarp. Pericarp means outer covering. Okay, such has been described inside the picture. So if you did not understood in this class, so in this class is very fast, this video may be very fast, so if you did not understood, we will give you the source, so you may read it later. Okay, so this was the saffron staining on the seed of your mature seed of your monocotyledon seeds. Okay, so in this seed you can clearly visible the part of your coleoptiles and the rest of your part that did not take place in their germination, that is the endosperm, and which helps you for the nourishment. Okay, and the scutellum, which was the cotyledon, form a single cotyledon from your monocot seeds. Okay, and the premiole is the part where the shoot apical meristems and the shoot primordia are formed. Okay, so it is the radical part inside your monocot seed. So the covering that coated your radical or root tips, that is your coleorhiza, and the total pericarp, here the pericarp has been seen, it is the seed coat. Okay, the seed coat fused with your pericarp, the outer covering like your rice upper yellowish part of your covering of your rice is your pericarp. Okay, so in this picture we can identify the following part of our seed, that is the number 1 point. Okay, mark the number 1 point is your modified cotyledon of your complex monocots such as grass, and the coleoptile. So in the second part, the first part is your scutellum and the second part is your coleoptile. So in the coleoptile, the sheath is covered by the young shoot, that is premioles, and the coleorhiza, which is your fourth part is your coleorhiza, which covers your roots. Third part is also coleorhiza. So fourth is your premioles, and fifth part is your endosperm. It was endosperm, and sixth part is your which fills the most of your part of your seed and nourish the seed upon your germinations, that is your endosperm, and this part is your endosperm. Okay, that may be messy, so तुमको मो ओडिया रे बुझे दोची. तुम सीड रो एई पार्ट हला तुमरो कोटिलेडन. कटिलिडन न अपर पार्ट जो फॉर्म करछि प्रीम्यूल फॉर्मेशन करछि प्रीम्यूल रो अपर पार्ट जो कवरिंग टा रहिला दैट इज योर कोलेप्टाइल ए पार्ट होछि तमर रेडिकल जो कवरिंग करिक रहउछि दैट इज योर कोलियोराइजा राइजोइड फॉर्मेशन कोलियोराइजा ओके कोलियोराइजा हे गला एंड रेस्ट ऑफ योर पार्ट जोटा अछि सेटा कोन तमरा रहि गला एंडोस्पर्म पई ओके एंड एटा जो कवरटा रहि के अछि 7 नंबर ऑफ पार्ट होछि तमरा दैट इज योर पेरिकार्प एंड सीड कोट दैट कवर्स योर सीड ओके सीड टा कवर करके रखि छी आई थिंक माने बुझी साथिबो so some of species may be seen the variation that seed are diverse and like a pinus that is a gymnosperm and thus neither the monocot nor the eudicote pinus ता तमरा monocot जीवनी कि eudicote जीवनी काहे कि सेटा हम कहबी बिकॉज़ सेटा कोना pinus तुम जदी सीड्स ता देखिथिबो सो से ए टाइप स्ट्रक्चर होइथबो बिकॉज़ ता सीड भीतर ही सीड थाए ओके and some plants like orchid that is orchidaceae is a monocot but do not develop as a embryo and even endosperm in the seed and their germination depends upon the presence of your symbiotic mycorrhiza fungus in case of orchid the presence of your mycorrhiza fungus will help so germination of your orchidaceae family species so तुम देखि परिबो so difference कोन रहउची ना चीमेज साप्थोग्लोटिस क्रिम्बेडियम्स बाइकलर ये सब कौन ना तुम मोनोकटिलिडन्स रे रहले ओके डाइकटिलिडन रे तुम कौन रहले ये लिगियम फैमिलीज रो रहले स्वाम्प वाटर्स रहले गोल्डन सेंस रहले एंड सोयाबीन्स रहले चिकपी रहले एंड गार्डन नस्टिरम रहले ओके सो सेमान भितर एम्ब्रियोनिक डेवलपमेंट रे चेंजेस रही थाए सब गुडा कर देखिथिबो एम्ब्रियोनिक डेवलपमेंट कोटर ग्लोबुलर रहला बेले कोटर कोन ना कोटर ग्लोबुलर रहला बेले कोटर इलोंगेटेड स्ट्रक्चर होची कोटर टॉर्पिडो लाइक स्ट्रक्चर होची कि कोटर रेडिस लाइक स्ट्रक्चर होची ओके एई टाइप स्ट्रक्चर से देखुची दैट बिकॉज ऑफ योर इवोल्यूशनरी चेंजेस इनसाइड योर जीन चेंजेस रे इवोल्यूशनरी चेंजेस रे सेटा को हमें केमी देखिबा ना एटा देखो तुमरो एटा होची पीरियोडिक ट्री होते तुमरो फाइलोजेनी जदी तुम पढिथिबो एटा होची कोन ना तुमरो पीरियोडिक ट्री रे फाइलोजेनिकली तुमको स्पीशीज गुडा को डाइवर्स करा जाईछि सो फर्स्टली हमर कोन ना ब्रायोफाइट्स सब लोअर प्लांट स्पीशीज गुडा कर जाई थांती सो मॉस और लिवर वर्थ एंड हॉर्न वर्थ गुडा करे से मानकर भीतर को टाइप स्ट्रक्चर होइथबो ए टाइप स्ट्रक्चर होइथबो ओके एंड लाइकोफाइट्स लाइकोपर्सिकम्स एंड क्लम मॉस एंड स्पाइक मॉसेस गुडा करे ए टाइप एम्ब्रियो स्ट्रक्चर होइथाय ओके like this type of embryo structure होइथांती and monilophytes that is form मानकर embryo गुडा को ए टाइप रो फॉर्मेशन करथांती बट इन केस ऑफ जिम्नोस्पर्म रो पाइनस से देखो ए टाइप रो स्ट्रक्चर बनै थांती बुझिला तार embryo टा ए टाइप रो होइथांती बट इन केस ऑफ एंजियोस्पर्म रे हमरो केमिते होइथाय दैट इज ग्लोबुलर होइथिबो देन सेटी कोन ना एटा होची तमर कोन ना सस्पेंसर पार्ट सस्पेंसर डेवलप होइथिबो देन दैट विल फॉर्म एज योर टॉर्पिडो स्ट्रक्चर होबो ओके ए टाइप ऑफ स्ट्रक्चर टा बनौची सो तुम माने डिफरेंस जानि सरथिबो ओके so I think you may have clear the concept about what are the monocots and what are the dicots so in this picture also here it is the total embryonic embryogenesis or embryo development of your dicot seeds and it was the development of your monocot seed okay so Arabidopsis embryo that is mustard embryo is firstly develop into zygote then one cell that is formed the apical cells and basal cells then apical cells then develop into 2 cells, and basal cells develop into 4 cells. That is 2 to 4 cell stage. Okay, and then it goes to your dermatogen process. Maybe in this case your seed dormancy maybe takes place or not, may not. Okay, after the dermatogen process, the globular stage has been seen. Globular stage means your globule like formations. Okay, and SU means your suspensor. Okay, then at the globular stage, after that it forms a heart shape, and then the torpedo stage, and then it bends and forms a mature ovule, mature seed structures that was not purely mature. Then it forms your mature cotyledon structure which was bifurcated. Okay, but in case of your gaminous embryo, gaminous species रे कौन monocot species रे seed node zygote develop into apical and basal cells. Then it was forms the suspensor, and the suspensor was merged with your apical cells also, and that forms such like structures. And then it forms your root apical meristems and shoot apical meristem. In case of your shoot apical meristem, the cotyledon that forms the shoot apical meristem, it was known as your scutellum. This part is known as your scutellum, and it was the female in which your shoot apical meristem presents, and apical primordia also present. Okay, so okay, I will discuss up to 2 to 4 side. It may be a lengthy class, so you may not able to understand the topic. So angiosperm embryo develop, how the angiosperm develop in this picture. It was the egg. We all know that after the fertilization and the process in which egg forms the zygote, and the triplet fusion product is from your endosperm. In the first division, at your zygote's first division, it forms into your apical cells and basal cells. Okay, apical cell and basal cells. Apical cells further divided into 2 cells, and basal cells divided into 4 cells. That forms your suspensor, and then this structure is developed into your globular structure. Then it was developed into your heart shape structure, and it was developed into your torpedo structure. Okay, in case of a torpedo structure, the cambiums and procambiums. Cambiums means the presence of your cambium like structure that due to the presence of your phloems and xylem, okay, develop inside your seed, which helps you for the transport of your waters and the nutrition elements to the seed. Okay, that develop inside your torpedo structures, and then it forms the mature seed structure. In case of your mature seed, the cotyledon was purely developed, and shoot apical meristem also developed. Okay, during the germination, the both cotyledon will be growth, and that helps to the absorbing of nutrition, and the shoot apical meristem, this portion will be then increased to form the stem and seeds, stems and leaves. Okay, I think you understood. This was the complete development stage of your Paullinia. Paullinia species रे तमर केमिति कोन डेवलपमेंट होची सेटा भी तमर ब्रीफ डिस्क्रिप्शन थिला, तो so that I propose this picture, so that same as of your general dicot embryogenesis process structure. So it's the different type of stain they have that, so that the cotyledons and sham, that is shoot apical meristem, also seen clearly colorful. I will discuss it later in this slide. Okay, so this was the process of your embryogenesis in your dicot seed. Until this process, the zygotic genome activates that maternal and parental factors emergence and the epigenetic reprogramming. Okay, epigenetic reprogramming until this process, and the pattern of formations was followed by some signaling pathways that are like a SSP YDA, MAP kinase, signaling cascades, and oxygen synthesis. These are the factors. Oxygen may also helps in the seeds germinations. Okay, and some the proteins that are helps for the root apical meristems development and subapical meristems development. Like these are the genes, that is OX5 genes that help for RAM production, and transcription factors such as transcription factor, mRNAs, and inner and outer transcription factors also helps in your germinations. And subapical meristem transcription factor that helps for your cartilage development and the germination process of your shoot apicals. The shoot primordia forms by the transcription factor, and specific type of transcription factor also act in the process. Okay, and vascular tissues that forms the cambium and procambiums in the epidermis regions. And in the at the maturation stage of your seed, that formation of your chlorophyll, so that the seed are look like a green color of your both the cotyledon, and tag synthesis protein accumulation at the 20s globulin and 20s globulin. Sorry for the disturbance. And after the maturation, the cell division and expansion also takes place, and desiccation and dormancy transcription factor also act. Okay, so this is the development of your monocot embryo. You may know that how the dicot embryo also form. Such as the such process also takes place inside your monocot embryo, but the cotyledon not forgotten, that presence of a single cotyledon. Let's discuss how it formed. Okay, firstly your oospore has been present, that is your zygote that forms the apical cells and basal cells. Okay, then sorry, this was inverted. This was the apical cell and this was the basal cells, and basal cells then forms your suspensor, and the apical cells formed continuously forms the cotyledon streamers, like the part which will grow. Okay, at certain stage it was the apical cell further divided and forms the primule and cotyledons. Okay, and that later on forms your such type of structures, that the suspensor was stock like substances that helps to the complete seed, the complete zygote to adhere. Okay, and that later on forms the structure like this. Okay, this was the typical monocot seed structure in which the scutellum has been present. Scutellum is your single cotyledon present inside your monocot seeds, and the cotyledon covering was known as your coleoptiles. Okay, and the presence of your shoot apex inside your cotyledon that presents at your apicotyl region, and at the below of it the hypocotyl region is also present, and the presence of your epiblast region, that is vascular bundle sheath containing part of your seeds, and radicle, that is your root initial part of your seed that forms as your root and root tips, and coleorhiza is the part where which covers your root tips and radicle. Okay, so ये गुड़ा पार्ट गुड़ा तुम्हारा मोनोकॉट एम्ब्रियो रे रहिया को मिलता है. सो आशा करछि तुम माने डिफरेंस बिटवीन मोनोकॉट एंड डाइकॉट with the जानी साथिबो. Okay, what are the difference? So this was the picture of your embryogenesis. We will discuss the embryogenesis process one by one later on. So how the globular embryo forms, and what are the functions, and heart shape and top to state, what are these structures functions and its involvement in your formation of your seed. Okay, we will discuss it in the next class. Okay, thank you.